Welcome to Lecture 15, where we will go over the cell cycle, or the process of cell division. Remember, according to cell theory, all cells derive from each other. As a result, cell division is absolutely crucial for cellular reproduction and for the continuity of life. There are many purposes to cell division, such as reproduction, which is particularly important for unicellular organisms to reproduce. Cell division is also important for growth and development and for tissue renewal. In the process of cell division, typically two identical daughter cells are created. This is a highly coordinated process that is controlled through the use of the cell cycle. It is important to understand the overall structure of DNA to understand how the DNA changes during the cell cycle. Remember, the genome is considered to be the full complement of a cell's DNA, so that it contains all of the genes that an organism has. This genome can be in one of two forms. It can be in the form of chromatin, which is the unwound form of DNA. And remember that this unwound form of DNA will also have small proteins attached to it. This will help to fold up the DNA when necessary. The DNA can also be present in chromosome form. This occurs when the strands of chromatin become highly condensed. This is the form that chromosomes or the DNA takes when a cell is actively dividing. Remember, when a cell is normally functioning as it should be, the DNA is going to be in the form of chromatin. This is because it needs to be transcribed and translated into proteins on a regular basis. DNA can differ with respect to the prokaryotes and eukaryotic cells. In prokaryotes, the DNA is very simple and housed in one circular chromosome. However, in eukaryotic cells, because they are more complex in structure, eukaryotic cells have many linear chromosomes, and these chromosomes are going to be housed in the nucleus. Eukaryotic organisms typically have two different cell types, either somatic cells or body cells. These consist of all of the different cells in the body, except for your gamete or germ cells. These somatic cells can be termed diploid because they have two copies of the genome. In humans, we have 46 chromosomes in total. 23 originated from our mother and 23 originated from our father. Eukaryotic organisms also typically have gametes or germ cells. These would be consistent with the sperm and the egg. These gamete cells are considered to be haploid because they only have one copy of the genome, meaning that in humans, the gametes would have 23 chromosomes. This image here depicts the nuclear DNA within the nucleus. We can see that we have many linear chromosomes that are present within this eukaryotic cell. As stated previously, chromosomes will only form when a cell is preparing to divide. This is because it's going to have to neatly package its DNA so that it doesn't become damaged during cell division. The DNA is going to be duplicated, specifically in the S phase of the cell cycle. After the DNA is replicated, each chromosome now has two identical sister chromatids. This is considered to be consistent with the original DNA chromosome and its copy. Once DNA has been replicated, the cell will then enter into the M phase. The M phase is when these chromatids separate and eventually are passed on to the two daughter cells. The M phase refers to mitosis, which specifically refers to the division of the nucleus or the division of the genome. Mitosis is then followed by cytokinesis, which refers to the division of the cytoplasm, which will eventually lead to the existence of two daughter cells. We are now going to take a look at the various stages of the cell cycle. Notice that the cell cycle consists of many other stages aside from mitosis. Mitosis simply refers to the actual act of cell division. So the phase of mitosis is actually going to be very, very short in relation to all of the other phases. The start of the cell cycle begins with the G1 phase. This is the first gap phase. It is typically one of the longest phases of the cell cycle. In this phase, the cell is undergoing a large amount of growth. It is increasing its cytosol, it is increasing its plasma membrane, as well as increasing the presence of organelles. This is because one parent cell will have to divide into two daughter cells, and these daughter cells will have to have all of the machinery necessary for them to survive. After the G1 phase, the cell will enter into the S phase. This is the phase where DNA is going to be copied. So we call this the DNA synthesis phase. It is also important to note that in the S phase, the centrosome, which will help eventually divide out these chromosomes, is also going to be duplicated in the S phase. This is because there's only one centrosome present in a parent cell. In order to produce two daughter cells, they each need to have a centrosome as well. So at this stage, you're going to duplicate the centrosome so that cell division can occur properly. Once the S phase is completed, the cell will enter into the secondary gap phase, or G2. Once again, this is a growth phase. So organelles are going to be duplicated, mitochondria are going to replicate, organelles are going to be produced, cytosol is going to be produced, as well as excess cell membrane, in order to ensure that both daughter cells have the sufficient machinery to function. Cells that are not growing or dividing are considered to be in the G0 phase. The G0 phase is an important phase for any cell that is not dividing. This means that a liver cell that is functioning as a liver cell is going to be in the G0 phase. It is only when it needs to divide that it will enter into the G1S or G2 phase and eventually undergo mitosis. Many cells can be actively dividing, such as your skin cells. Skin cells need to be replenished at a very high rate. So as a result, these cells are going to be entering into the cell cycle on a regular basis. So their time in the G0 phase is going to be limited. However, other cells, such as your neuron cells or brain cells, are going to be locked into the G0 phase for an extended period of time. They're not likely to divide, and so as a result, they're going to spend much of their lifespan in the G0 phase. Interphase refers to basically all of the cell cycle stages except for mitosis. So that means that the time a cell spends in G0, G1, G2, as well as S phase is all going to be considered their time spent in interphase. We can also see from this diagram that after mitosis is completed, we will have cytokinesis, which once again refers to the division of the cytosol. At this point, you would have the production of two daughter cells. We're now going to take a look at the specifics of mitosis, the process of mitosis, or the M phase. In mitosis, we have the nuclear division. This is important because we have the production of two identical daughter cells. The process of mitosis is common for your somatic cells. Remember, we're going to start off with a parent cell, or somatic cell, which is diploid, and we need to produce identical daughter cells. This is done through the process of mitosis. Gametes undergo a different process called meiosis, which we will take a look at in the next lecture. We have 5 general phases of mitosis. They consist of prophase, prometaphase, metaphase, anaphase, telophase. This will then be followed by cytokinesis. Remember, before a cell enters into the phase of mitosis, it has gone through interphase, which means it has gone through G0, G1, S, as well as G2. This means that the DNA has been duplicated. This also means the chromosomes are present in the form of chromatin, and they are usually found inside of the nuclear envelope. Remember, we also have the duplication of the centrosomes, because those are going to be responsible for pulling apart the chromosomes. However, these centrosomes are still paired together, and they will eventually need to move to opposite ends of the cell. We are now going to take a look at the details of prophase. In prophase, the chromosomes condense down and become visible. This means that the DNA is going from the form of chromatin into the condensed chromosome form. In prophase, you can also start to see the sister chromatids. This is because the original and associated copy of DNA are going to be held in close association with each other. During prophase, the nucleoli also disappears. Remember, the nucleoli or nucleolus is simply a dense region within the nucleus where we have the ribosomal transcription occurring. So ribosomal RNA and the ribosomal subunits will begin to form in the nucleoli. However, we do not see this in prophase because the ribosomes are not going to be produced at this stage. We also have the formation of what is termed the mitotic spindles. This is where the centrosomes, which are, remember, microtubule-based, they're going to start extending out microtubules that will eventually connect on to the chromosomes to help pull them apart. We can see asters appearing within, or star-like structures appearing within the cell. You can see this in the diagram to the right. The green structures, which are reaching out, represent the microtubules, and the centrosomes are centralized within the base of that structure. In prophase, the centrosome pairs also begin to separate. This is because you're going to need those centrosomes to be located on opposite ends of the cell. In prometaphase, the nuclear envelope begins to dissolve. This is because the nuclear DNA is going to have to be accessible to be able to separate out properly. So as a result, the nuclear envelope will need to be dissolved. In prometaphase, the chromosomes become fully condensed. And now we can see what is called the kinetochore at the center of the centromere. The kinetochore is going to be the eventual location where those microtubules are going to attach to the chromatid to be able to pull them apart. The microtubules of the spindle that are coming from the centrosome are eventually going to invade the nuclear area, and they're going to start to attach to the chromosome, specifically at the kinetochore. We can see in the diagram below how that happens, with the microtubule extending, joining into the kinetochore, and connecting to the chromatid. We now enter into metaphase, which is considered to be the longest phase of mitosis. In this phase, the centrosomes are going to be aligned at opposite poles, which we can see in the diagram below. We have the centrosomes originating at the top and bottom of the cell. They also have their microtubules extending towards the center to attach to the kinetochores of the chromosomes. In metaphase, the centromeres are going to be lining up on the metaphase plate. This means that all of the chromosomes will also be aligned along the metaphase plate. This is an imaginary area within the cell where the chromosomes are aligned. At this point, all of the kinetochores of the sister chromatids are going to be attached to opposite spindle poles. This means that for each chromosome that you will have its original as well as its copy in the form of sister chromatids. So within each chromosome, each chromatid is going to be attached to microtubules that are going to be either attached to one centrosome or the other. This is to ensure that when those chromatids are pulled apart, each one is pulled apart to opposite ends of the cell. We say at this stage the spindle apparatus is fully formed. This means that the centrosome has now fully grown out all of the microtubules, and all of these microtubules are fully attached to the centrosome. At this point, we are now ready to start pulling apart the DNA to eventually form two daughter cells. We now enter into anaphase. This is one of the shortest phases of mitosis. This is a very important phase because this is where the sister chromatids actually separate. They are going to be pulled apart by the spindles and eventually start migrating towards opposite ends of the cell. When the chromatids are pulled apart, the microtubules will shorten, pulling the chromatids apart as they shorten. At this point, because the original and copy of the DNA is separated, We now can term each of these chromatids an independent chromosome. At this point, the cell begins to elongate to eventually form the two daughter cells. We now enter into telophase and cytokinesis. Cytokinesis begins in telophase as well, so you cannot separate out these two phases completely. In telophase, we have the two separate daughter nuclei that are beginning to form. This means that the DNA has been separated. The chromosomes will start to unwind and go back to chromatin form, and you're going to have the formation of the nuclear envelope to help protect that DNA. You will then have cytokinesis take over, which once again begins during telophase. When cytokinesis begins in eukaryotic cells, you can typically see what is termed a cleavage furrow. This is going to be eventually pinching the membrane off to form two cells. A cleavage furrow will also be present in the division of prokaryotic cells as well. These slides simply depict the different phases of the cell cycle. We start with G2 of interphase, where we've got the centrosomes duplicated. We can see in the diagram below the first part of the cell. And remember, these centrosomes would have been duplicated in S phase. We also see that the chromosomes are duplicated as well, and they are present in the chromatin or uncondensed form. Because we are in G2 phase, we still have the nucleolus and nuclear envelope. Entering into prophase of mitosis, we now start to see the mitotic spindles forming, which simply means that microtubules are going to start growing out from each centrosome. We're also going to have the separation of those two centrosomes, and they're eventually going to start moving towards opposite ends of the cell. We can term the centrosome and the microtubules extending from it as an aster. In prophase, we can also start to see within the nucleus that the chromosomes are starting to condense. If you take a closer look, you can see that there are two strands associated with the chromosome. One would be the original DNA chromosome, and the other would be the corresponding copy that was synthesized in S phase. Hence, the chromosomes in this form have a typical X-shaped form. And here we would refer to them as two sister chromatids. This is because they contain the exact same DNA. Each chromatid, whether it's an original or a copy, is identical to the other. In prometaphase, we can see that the spindles are now at opposite, and the centrosomes are at opposite ends of the cell. We can also see that some of the microtubules are connected to one side, and sometimes two, of the chromosomes. And at this stage, the microtubules are extending to start pushing the chromosomes towards the center of the cell. This is so that they could eventually be pulled apart properly. We can also see that the nuclear envelope is starting to dissolve. This is so the microtubules can extend through and access the DNA. We also see the formation of the kinetochores, and this is where that provides that attachment site for the microtubules to attach to the chromatid. We can see at metaphase that the nuclear envelope is fully dissolved. We also have the full extension or fully formed spindle fibers, which means that the microtubules that extend from each centrosome are going to be fully formed. As a result, the chromosomes are going to line up on the center of the metaphase plate. And please also note that each chromosome will be attached to each spindle fiber, which means that for each chromatid within a chromosome, they're going to be attached to a microtubule that eventually will pull them to either side of the cell. In anaphase, we now have the separation of those sister chromatids. As the microtubules shorten, they will pull those sister chromatids apart and pull them towards the respective poles. Then we will have telophase and cytokinesis, starting with a cleavage furrow forming that will eventually pinch off and form the two daughter cells. We can also see that the chromosomes are now fully separated and starting to unwind. As they unwind, we will get the reappearance of the nuclear envelope. Also note that within each daughter cell, we now have one centrosome present.